What I'll try to present to you today is some of the work we've been doing on systems biotechnology since the, let's say, early 2000s, late 90s, and some of our ideas on how it will progress. Eventually, some of the things I'll be saying will be a bit arrogant, typical of engineers, but at the same time, you have to realize that one of the main reasons why we've been doing them for a long time is because the engineers realize our brain is small, we are lazy, and so we cannot cope with the complexity of biology. Thus, we need tools, in particular mathematical tools, to organize our brain and to tell us where the peaks and the valleys are, and hopefully those models will also tell us not the solution. There are no model that can give you a solution at the moment in this area, but at least where north is, so that you can kind of progress through the research you do and gain from it. And in particular, we've been working in this area, as I said, from the late 90s, in, for example, gene therapies or areas close to cell therapy, but of course also vaccines. We've been long past the day of proteins. Proteins are now much more of a mature area. And through the difficulties we found in those areas, which were more artistic than scientific, slowly the science has been establishing itself, we try to somehow wrap them around processes in which we can improve the understanding. And finally, it's our feeling that sooner or later we'll shift paradigms. We all know that so far the process defines the product. The more you'll know about the way in which the cell and the environment work, the more you'll be able to transfer to the new paradigm. And of course, we are in the early days, so a lot of our models are still, okay, no longer that easy, but still very primitive, because there is not enough knowledge to incorporate. But, and that's the arrogance of the engineers, in the past we had similar situations. When we were developing, say, polymers in the 30s and 40s, we only knew some of the kinetics, we only knew some of the stoichiometry, and still we could put process to work. So it's the same kind of arrogance, if you want, applied to a much more complex system than the strict chemical system is. And so essentially, our understanding, of course, and this is from a paper which Nuno Carinhas published some time ago in Trends in Biotech, the available tools which you have for systems biology are generally coming from the large-scale, not particularly dynamic, non-parametric, all the way down to the parametric ones. And then you have on this, the Y-scale, the modeling methods you can use, and some of the experimental methods that have been used throughout. The idea being that once you start to put the correct picture with all the tools you have at the moment, you'll be able to progress further and at least make some hypotheses which are based on more or less sound knowledge. I'm going to give you some examples, and some of them are more historical than others, of how we've progressed this road in the IBET lab, and those are including the areas which are quoted green there. The first block of things that were put together relate to metabolic characterization, modeling, proteomic analysis, and this in particular is for the baculovirus system production, where we've been working for many, many years, both with inogenetic and in the days when GlaxoSmithKline and Rix and Sart were starting their work in insect cells, that finally led to them producing the human papillomavirus vaccine in a Hi5 cell. So we've been with this trend for a long time, mostly to produce proteins, but more recently also to produce baculoviruses themselves as gene therapy vectors, which are now under use by ARC, for example, in England and in Finland, and a few other companies. So I'll be reviewing some of the work we've done there. Then we will jump into the metabolic behavior of culture mammalian cells, so mammalian cells themselves. We are being old means that we started in the late 80s on monoclonals, and in the late 80s the fight was not decided yet between BHK and CHO. What happened was that in Europe we were working BHK with Merck, Germany, before they then bought the Erbitux from the States. And we were developing a monoclonal in BHK, and I'll present some data using that same cell. Then, of course, I'll also present some data on CHO cells producing monoclonals out of the work we do for Lonza in fluorometric control of fermenters or fluorometric detection of 96 well plates to get to pick the best clones and things of those sorts. So What happened then and throughout this process is that, as I said, our brain not being good enough, we tried to get models to allow us to go the right way, hopefully. And finally, I'm not so sure how much time I'll have for the rotavirus systems and all the mathematics behind the VLP assembly and the dynamics of the stoichiometrics and dynamics of the stuff. If I get there, I'll get there. Otherwise, you'll have it in your presentation. I'm very happy that I can talk about insect cells today because about 3-4 weeks ago, finally, we have an SF9 product, not an I5. I5 is the GS-CHO, an SF9 product, the flu block, coming from Protein Sciences in Connecticut by a lady who, for those who are old enough, a lady who got a PhD in Wageningen with Monique von Hors, who was an ex-student of Just Vlak, who was one of the first guys doing really the molecular biology of SF9s. And that lady is Manon Cox. She is the boss and now very much in the news because flu block was approved for influenza a few weeks ago. So the rest you know, it's a well-known system for preclinical type of work and everything that is needed there. And when we were starting to work on this, we realized that there was a cell density effect in baculovirus replication. That's been known for other types of systems. For those of you working in adenoviruses, for example, this same problem existed a few years ago. And of course, we and others have somehow solved it. What I'll be talking about here is the particular one of the baculoviruses because it was more recent, so some of the tools I want to put forward to you have been used here. What does this mean? It means that as you increase, as engineers, what you try to do is always the same. Increase the number of cells in the system, either because they're excreting something you want, or in the particular case of a system which has a second step of infection, assuming that by the time you infect more cells, you'll get more product. What happens is that, as I said, for the adenovirus system, at least in HEC293 cells, that was the case. It's not so much the case in the CEVEX cell lines, on which we're working as well for the adenoviruses. But here we found the same situation when trying to produce baculoviruses. If you increase the concentration of cells at the time of infection, each one of the cells will produce less viruses. And of course, you have to find out what's happening, because at the end of the day, what you need is the volumetric productivity, but if the cell-specific productivity has gone down, the advantage of having more cells is probably not going to be that high. So we were looking at it with the old tools, or more or less old tools, and of course trying to say it was more or less obvious that it was not the typical things from mammalian cells. Insect cells are rather different. They have no ammonia problem. They have no lactate problem. So looking at them, it didn't look like it was the usual situations coming from the animal cells, so we had to try to do the usual metabolic flux analysis, typical of engineers, right? In this particular case, 30 whatever types of equations, looking at them, and then doing some manipulations to check what was ongoing. And the point I'm trying to make, if I can make this pointer to work, is about here. So what you have is, in terms of the energetics of your system, cell concentration at infection 1 million, 2 million, 3 million. That's what will be the readings there. And what you have is a situation that it looks as if there is a depression of energy generation following infection at increased cell densities. So this is old story, it's published, I'm only giving you the final results. We've of course also looked at the enzymes involved, and we found out that if you either plug pyruvate or alpha-ketoglutarate in your system, you're able to get final results of this sort, i.e. the graph at the bottom is a cell concentration at 3 with other amino acids. There's nothing really happening. With those two, which we identified, you are almost at the same level at what you get at cell concentration 1 under normal situation, infective particles per cell, which drop dramatically when you go to 3. And so in final titer, in infective particles per milliliter, you get with alpha-ketoglutarate a substantially higher amount, 3 times more than you would get on the other system. So in a way, this is a potential solution for this case, and that's how we went. Historically, we jumped then into areas where we started feeling, okay, some of the same systems you've used in chemical engineering before may be ripe to be used here. And this is, again, old stuff. First thing we published on this was 2007. What we do is, to some extent, repeating what we had before. Everything on which we know data, equations, stoichiometry, even kinetics, which is the mechanistic component, we write everything down. Of course, in principle, the idea is that by knowing all this, if you plug in something there, you should know what will be the product coming out, tighter, whatever. But unfortunately, there are a lot of things which you don't know, in particular regulatory and others in your system, and that part is then unknown. So here what we did originally was developing partial least square approaches. Multivariate analysis was one of the drugs there, trying to correlate a few manipulations. And in principle, if you do this, you need to do a lot less manipulations than in a typical design of experimental system. And since we're lazy, that's what we want to do, the least manipulations we can. Then, of course, each manipulation, we have to live in a data-rich environment because we have to analyze everything. But it gives us tools that will allow us to know which way to go and how to go. So, mechanistic component, metabolic flux analysis is no major problem. Then you have a sequence system where the partial least squares integrate what you've measured, and you go back and repeat your system until you get a decent system from the mathematical point of view. And what you have here is we've used some data for calibrating the model. That's black dots. And then, of course, you have to add other data to see what your predictions will come out correctly or not, and that you have to do again. So the validation correctly falls into the predicted model. So we have in our hands a list of rules to develop metabolic and bioprocess engineering strategies. As I said before, it doesn't tell you what the actual title will be, but it tells you if you follow this route is a lot better than if you go somewhere else and you have a few of those lining up. And of course, this kind of mathematical basis has been used already for things like looking into how you go into the cascades of signaling throughout the cell. Don't go that way, go somewhere else. So this is expanding in other areas. We're doing nothing which is totally new. It's only the fact that we're applying it to this particular case and getting out of it. And then, of course, what you do finally, and this is more recent story, but similar to the old case I've shown you before, you go through all these regression analysis, identifying the fluxes and the numbers, and then doing the statistical filtration, and you come down with essentially two respiration and the TCA cycle, which are the key determinants of the baculovirus production as a viral vector, and this is similar to what we had before. So in a way we have, if you want, a more basic type of description of what we had already found through more circumvoluted ways. And of course, you can also do it and get better understanding of what's ongoing for you if you jump into the some of the proteomics which are becoming available. This is, again, something that's been rather forgotten. Everybody knows already a lot. In the CHO case, at least, we already have a big genomic effort, but the proteomic is still in the early days, and not much has been done before. And of course, most of it required to go through the gel patterns until not that long ago at Max Planck, they started putting together tools which are called, there are other tools, but they are based essentially on systems which have labeling, and the one we use now, and that's probably the strongest for us, is the stable isotope labeling of amino acids in animal cells. And essentially what it means is that with mass specs, you can distinguish things which only diverge by, say, 2, 4 Dalton, 6 Dalton. And what you do then is that you have labeled stuff, arginine or lysine labeled stuff, as opposed to the rest of stuff. And then if you now design your experiments correctly, and if you have the mass spec systems to cope for it, you can get the indications of what happens when you have low cell density infection, high cell density infection. Again, we're still looking at infection, not so much growth. Of course, we put growth in as well because the experiment was having to go through growth anyway. And by designing the experiments properly between the labeling and non-labeling, and of course doing the deconvolution of the mathematics, you come out with maps like the heat maps you have from the genomics, which show you what's happening for some of the enzymes which are more relevant for you. In this particular case, since we don't have the whole protein map of the SF9 cells, we had to go through other libraries from, from Drosophila to I5 to whatever to get a decent map to create. But this is just the final result. So again, you have an indication of what are the probable more relevant targets for you to pursue in the following experiments, which is you in the system. So another omic tools. And of course, out of this kind of a presentation, you can both have some clues on how to improve the viral manipulation and at the same time how the cell density has been affecting your system. And once you know what's happening, you can much better go and find a solution. Some cases maybe you won't find one, but at least you know what the basic understanding is. And as an engineer, I have absolutely no doubts that there is nothing as practical as a good theory, in the sense that if you really know the subject, eventually you can solve it. If you don't, okay, you're shooting in the dark. So this is the kind of work which will continue in our lab and others to allow us to go through predictions which are better targeting what really will be interesting from us. Of course, once you start doing that in a lab with very good students and some interesting problems and some pieces of equipment around and other people doing many of the other things, you start looking at, will this be of any value? And very often, some of those things come out of talks with people like those in this room. This was a kind of a discussion with some of the, we've been working with for a long time with the German Merck nearby in Darmstadt, now Merck Serono, but we've also been working for more than 20 years with the what we call the daughter Merck, Merck & Co., Merck's ST, whatever you want to call it, in the States. And this was coming out of talks with those guys, the Barry Bucklands, John Honest, and God knows what. They were saying, oh, you have such good tools in your system on the NMR. Sooner or later, this will probably be of value. Just put through some kind of experiments that might convince your potential customers that there is something you can put forward by using this kind of tool, which we thought would not be very relevant because, as you probably know, NMR is not that good in terms of the fine determination of things. You don't do parts per billion in the NMR. But anyway, this is the kind of work which we carried out. And again, this is already done with the Lonza GS-CHO cell lines with some of their low producer, high producers. And of course, we were beaten, so BHK is no longer that important. We have to join the CHO bandwagon. There's no point joining the CHO bandwagon for genomics because everybody who's good already is doing it. We cannot add anything. Let's join the other bandwagon on the other omics. And that's more or less what we're doing at this stage. So being able to, in the early stages we are in at the moment, to get all these typical 100 and whatever types of equations. And finally, this is still work in progress, so don't quote it yet. And anyway, we're not giving you the numbers anyway. And for example, looking at some of the preliminary MFA results on what is the actual effect of butyrate, which many of you know is commonly used in some of those cultures in fundamental terms in what's happening within or inside the cell of the CHO line. And so this is how hopefully we'll be in a position to be of more value for people in this area. Of course, jumping into the more envirome metabolic type of behavior, what happens is that, and this was clear in one of the slides which Anthony Davis presented as he was describing how they cope with their systems on the 3D files. They had the physiology and the environment. And of course, it's always the same. We use functional enviromics just because everybody knows about functional genomics, and so we hope you can more or less understand what we're talking about, i.e. a situation where you have a multitude of pathways, of course, but at any given stage, not all of them are active, because it depends enormously on what the envirome is like. An envirome then is what? Oh, everything, a metabolite, substrates, God knows what, physical conditions, and those things we can measure, hopefully, and then again, try to correlate them with what's going inside the cell. And of course, this being more recent in our hands, we developed a somewhat new process where instead of doing, as I've shown before, the mechanistic and then partial list squares, and going sequentially, making the system learn as it goes. We lumped the two together and went into what we call latent pathways, projection to latent pathways system, which means we only have one system, which is trying to optimize the covariance between what you have in the envirome and what you have in the fluxome. Okay, I'm not going to go into the mathematics because I don't want to lose you. I probably have lost some of you already. But the final outcome is that by doing this, and of course, what you have essentially is then the inner model, as we call it, kernel or whatever. If you come from physics, that's what you would call it. Anyway, by having all that, and again doing only a few experiments, you're in a position to get two major outputs: the active pathways at a given envirome, and also the map of the correlation coefficients between the envirome factors and the pathway fluxes. So in a situation like this, what you can get is, for example, this is the application to the BHK I was talking about. What you can get is the rank. After all these manipulations I'm not going through, you have the rank of the particular equations and how they impact the pathways, sorry, and of course equations behind the pathways, and how they impact the added variance. And then you have a functional enviromics map to give you an idea of how to modulate the pathway activity. Of course, if you get it too complex, you're still lost, but if you make some zooming into the systems, again, you can get the kind of trends that allow you to go forward. What happened is, we have friends in many of those companies, and for example, through some of the ex-students working at Genentech, we tried to get the Genentech data through our hands to see how this model was going. What happened is that all their data is somewhere in different kinds of files, and they couldn't transmit it to us electronically. And they were kind of saying, Manuel, that's interesting, but we already know our system. And then we realized a very obvious thing. If you're doing something very pretty novel, don't try to sell you at mature industries where they already know, and so they don't have much advantage in using this. So we were going round, and then we're lucky enough on a heavy drinking session with some of the Merck, Sharp and Dome guys. They came up with, but this could be interesting from our glycophi. They bought a company named Glycophi years ago where they intend to do monoclonal antibodies in Phichia pastoris. We had some experience in Phichia years before with Berlex in California and EMD on the east side for single-chain antibodies. So we knew a bit about the system in engineering terms, how to control that kind of stuff. And so I said, yeah, why not? And so we did all this work with them. The work was done at IBET and GenIBET at our pilot plant, apart from the last one, which they did in West Point, because this was the strain, the actual producing strain, and they didn't want to give it to us for obvious reasons. It could be robbed or stolen. And that's exactly how it came out. So now we are experts on Phichia pastoris with hands-on data. Of course, we cannot tell you the exact data for their system, but most of this is horizontal anyway. So we can get to a point where we know where to fiddle around to improve things. And this is important for two things. For them, the critical point was, at the end of the project, they have a program, and the program is mathematically based. So if they now go into production, that program is used to control the production. If you do design of experiments, you don't have that at all. You have the best solution, but then you have to go back. Furthermore, we've proven, in this particular case, and they were pretty happy with that, that we could converge on a solution much faster than you would do on a design of experiment thing. But of course we realized this is for the big companies, not for us. So what can we do with that kind of tools? Okay, we can eventually support media development for the cells in which you already have all this information. And that's why this small company outfit from IBET is now expanding into CHO, because if you only do Phichia pastoris, you don't have many customers anyway. But by the time you have CHO, everybody goes into CHO for obvious regulatory safety kind of issues. You don't want to take too much risk. So that's why we're developing the other thing I've shown you before. So I hope I've been able to convince you so far that some of these approaches will make you get to the solutions faster. Of course, you need to have the base, the home base work done. If you haven't developed your mechanistic models, you're going nowhere. But once you have it, and of course the fact that we have it now eventually might be of value for you, this can be used for many other things. My last step, if I still have time, I'm completely out of time, I believe, is to tell you a bit on what we've been doing also using the rotaviruses as a model system for viral-like particles. And what we have there is 3 of the viral proteins, 2, 6, and 7. 7 is glycosylated, and we've been working with this for a long time because we were lucky to get all the information from a Frenchman from the CNRS team in CERN, Jean Cohen. Unfortunately, he died of a stupid accident in his lab, and he gave us literally everything and his brain. When we had problems, we could phone him. And that's the major problem for engineers. If you don't have, as a partner, people that really know the biology of the system we're studying, we lost because we asked the questions and the people cannot answer them or not even give us clues on what we should look at. So this was phenomenal. That's why we keep it in our hands, because we have everything from the tools, monoclonals, polyclonals, cDNA, blah, 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 all the way to the interpretation and the structures of the viruses and what kind of system you have. Of course, as before, we had to do a lot of work to go through the whole description, the whole mathematic description of what goes inside the cell as it's producing, even before the infection can be stochastic or deterministic, so you have to go through all these things and then measure the mRNAs, the mRNA decay values, and then the DNA, and then all these things. And so we had to develop methods, and this is the key point in a lab that really has to live in a data-rich environment. You need methodologies, analyticals, and I hope it was obvious also in the presentation from Margarita. If you're only saying your cells are viable, you don't exist. You have to show God knows what, from the up to the karyotype, genotype, or whatever, for a cell product. Here, it's a bit less exigent, but still you need to know a lot more than if you're putting out just a small product. So we've done all those things, and at the end of this, we came to a solution. I'm only giving you the integrated solution because everything else has been published before, where we have all the mathematics for this particular case. Of course, it will not apply. For example, the human papillomavirus is a 1-protein VLP. We are in a 3-protein VLP. So it will not exactly apply similarly to that particular case, but it will apply reasonably similar with the equivalent changes. We're using part of this mathematics that go all the way from the infection to the way in which you integrate the different viral proteins through dynamics and stoichiometry into there. What does it mean? It means that now I have, and that's the disadvantage of mathematics, I have a continuum of potential outcomes where I can go back to a molecular biologist and ask him, I need this protein to be there at this given time, and I need it to be there 3 times more concentration than the next protein that comes in later on. Because if this protein is in too high concentration, it will aggregate and get out, will never be used for the viral protein. If it arrives too late, it will no longer be incorporated with the rest. So I know what I want. Of course, if I then go to the molecular biologist, you'll tell me, oh, but I only have early, middle, and late promoters. So I don't have a continuum of solutions. But at least I can know that if I put an early promoter here and a middle there and whatever, I can get them in the right order. Not necessarily the right quantity, right times, but much better order to improve my titers. And as you know, synthetic biology will, in due course, give you, hopefully, promoters, synthetic promoters. So those things will develop with each other and will gain from those extra toolbox to our system once we know what we're asking for. Okay, this is going to the same thing, how they assemble, doing in vitro and in vitro kind of states, the stochastic modeling. It's not much relevant because, as I said, all of this, apart from the CHO work, has been published already, and if it interests you, we're obviously around, and Nuno Carinhas, who developed most of it, is close to Margarita, so he is the brain behind it. I'm only the salesman, as you probably realize at this stage. But the issue is, we think with all these tools, we'll be in a position to make better bets and go back to some extent to hypothesis-driven research as opposed to shooting in the dark, hoping things come. In many areas, it's too early. We're trying to apply this, for example, to vaccines. For example, in the model, one of the models that Margarida was showing, we've developed this. It's the brain model, the neural astrocyte cell lines. We already have all the mathematics of what we can get in the system, so we can do all the stoichiometries, not the kinetics inside the 3D systems. There, you need to sample and see what's going. But those things help you enormously in further pushing not only productivity but shortening the time that it takes to you to go for a go, no-go decision on different pathways of research. And this is essentially what I had to give you. Probably the photo is the same as Margarita, I'm not sure, but this is what we've been doing in this area. I hope I've convinced some of those more reluctant to use mathematics that we can use the mathematics if you can tell us what is your biology. So don't worry about it. The interface can be done. We've been doing it for a long time with lots of biologists, so long as the biology is reasonably well known. Thank you for your time.